Hello everyone, I'm Mike Ward and welcome to Conversations in Healthcare, a video series brought to you by DRG, part of Clarivate. This episode is one of a number that we are recording alongside On Helix, a digital conference that is hosted by One Nucleus. In line with this, I'm delighted to be joined by Sanjay Kakkar, who is a serial entrepreneur and is now CEO of a San Francisco Bay Area-based startup that's emerging from stealth mode, having just recently raised $30 million in a Series A round. Sanjay is CEO of Tranquis Therapeutics, a San Mateo, California-based startup that's focusing on the discovery and development of treatments for neurodegenerative and other age-related diseases. So Sanjay, I hope you and those you care about are keeping safe and well, and thanks so much for joining me.
Thank you very much, Mike, and good morning from California. It's really a pleasure to be invited to participate in this series, and thank you very much for your good wishes. We're all well, thank you, and I trust the same holds for you and yours.
Great. So, yeah, as I said in the introduction, Tranquis Therapeutics, it's emerged from stealth mode and it's going to focus on neurodegenerative diseases. Now, clearly that's a major unmet medical need that remains a major challenge for the life science sector. And it's certainly one that's been a challenge to resolve for a number of reasons, most notably struggling to identify clinically meaningful targets. So what is the target that Tranquis is homing in on, and who is responsible for the sort of discovery of that target?
Well, that's a great question, Mike. What we are doing at Tranquis is we're focusing on myeloid immune cell dysfunction in neurodegenerative diseases. Now, for many years, we've understood that neuroinflammation appears in neurodegenerative disease, and it was long considered an epiphenomenon. But Ed Engelman and colleagues at his lab at Stanford decided to focus their expertise in immunology. They had been pioneers in the field of immuno-oncology, to asking the question whether there are disorders of the immune system involved in neurodegenerative diseases that give rise to this neuroinflammation and are actually responsible for the pathophysiology of those particular conditions. So that's what they started to do some years ago, very quietly, started to look for pathways and targets that were downregulated or dysfunctional in patients with neurodegenerative disease. And then start to look for those defects in myeloid immune cells, both inside the brain, such as microglia, the normal cleanup, policemen of the brain, and also peripherally macrophages, because they felt that the peripheral immune system also had a role in these diseases. So that's what's really given rise to the approach that Tranquis has taken. It was incubated and identified in the laboratory of Ed Engelman and colleagues at Stanford, and then it became the underpinning of Tranquis when we span out of Stanford in 2007.
Right, and sort of looking at, for example, sort of the patent filings of that Engelman group in this particular space, they're looking at PPAR gamma cofactor 1 alpha, which is a sort of a target that was identified originally in 1998. And about 15 years ago, it was linked to sort of an association with some sort of degenerative changes in CNS. So, why has it taken that length of time from those sort of initial observations to actually move this into a potential target for neurodegenerative diseases?
Yeah, well, you're right that certain metabolic pathways have been identified, and also people look through GWAS studies to determine whether the genes regulating those pathways were dysfunctional in patients with neurodegenerative diseases. And that was identified, as you say, around 10 to 15 years ago. Originally, people looked for these dysfunctions and these defects in neurons and didn't really find anything. Now, it was Ed and colleagues, coming from an immunology perspective, started to ask the question, well, maybe the dysfunction that we know happens at a sort of epigenetic level in patients with neurodegenerative diseases is actually happening at a functional level in immune cells rather than in neurons. So that's what they've been quietly working on for some time now, some years, well before the company got going. And first started to, well, in parallel, started to look for the role of these pathways in myeloid cells in patients. And lo and behold, they found that the pathways that we are targeting are dysfunctional and downregulated in myeloid cells in patients with neurodegenerative disease, and very exquisitely so. But also started to look at doing knockout models of classic neurodegenerative diseases and knocking out the specific genes that we were targeting in myeloid cells in those animal models. And lo and behold, found that the animals had a worse phenotype of disease. So therefore started to prove that these were in fact important pathways and novel pathways that could actually provide a link between disease and an actual target mechanism. And so that's why it's taken quite some time, but we've made some significant breakthroughs quietly in terms of demonstrating that these pathways are downregulated in the target cell type, and that results in actually a disease process which can be targeted, can be modified, and where we can actually show we can restore the cell function back to normal. So in other words, bring it back to its normal function and take away its inflammatory state. So that's a double punch, really, and also show resulting benefits. And we've done that in multiple animal models of various neurodegenerative diseases, as well as develop an on-target biomarker because of the cell type that we're focused on.
So, I mean, one of the sort of big challenges that we've actually had in neurodegeneration is actually the lack of robust animal models that kind of give some indication of what happens in the animal model can be translated into the human experience. So what sort of confidence do you have in the animal models that have been developed so far?
That is a central aspect, one of the key foundations of how we designed and built Tranquis, the exact point that you're making, Mike, which is a very important one. So we've really started, because Ed's a physician by training, I'm a physician by training, we really started with the clinical problem. And as I mentioned a little earlier, what we did is first sought to identify in patients that we actually saw this response in terms of the myeloid cells, the myeloid immune cells, and that they were dysregulated. So that actually has become our target as well as our biomarker. So what we've been able to show in multiple animal models of neurodegenerative disease, which are challenging, but we have some highly encouraging data in models of ALS, frontotemporal dementia, Parkinson's disease, and age-related neuropathologies. What we've been able to show is that there is a common pathway, which is this dysfunction or downregulation of these particular pathways in myeloid immune cells, both inside the brain, namely microglia, and outside of the brain, namely macrophages. And that dysfunction actually tracks with disease. So with our therapeutic approach, what we've been able to do is restore normal function by upregulating certain pathways in those cell types. That results in those cells becoming less inflammatory, releasing less tissue-damaging cytokines, so therefore stopping their role in neurodegeneration, but also restoring their function back to normal, so they perform their normal cleanup function and can restore neuronal health. So by doing that, we were able to track directly between upregulating the pathway, effects in animal models. We're able to show in those animal models a reduction in the presence of inflamed myeloid cells, coming back to the mechanism. And we also already have an on-target biomarker where we've taken the blood of patients with disease and we've incubated that blood with and without our lead compound, and show we can restore the myeloid cell function of those patient cells back to normal in the laboratory, reducing the inflammation and restoring normal function. So therefore, we have an approach that we can bring into the clinic where very early on in patients, in very early clinical studies, we'll be able to track the relationship between dose, exposure, target engagement, markers of disease progression, and also clinical outcomes, which is one of the key ways that we're seeking to overcome, that graveyard that you mentioned, which has been very difficult in neurodegenerative disease research, jumping from the animal models through to some kind of predictability and target dose in clinic.
So, and I'm assuming that your drug candidate, is able to pass the blood-brain barrier.
Yes, our chemistry platform and our candidate molecules are already bioavailable and cross the blood-brain barrier, and we see them in the same or even higher concentrations that we see in the plasma.
Okay, so you just raised 30 million in a Series A. What did you tell your financial backers when you were raising that money? What did you tell them that they could expect to see you deliver with over the next sort of years or so?
Yeah, well, we're delighted with the raise. 30 million was the upper end of our targets, and we're absolutely delighted to have a sort of set of very supportive blue chip VCs in there. And so, they were all intrigued by, I think, two key things that we're seeking to build on in this Series A. One is this novel mechanism and the promise it holds to be a revolution in neurodegenerative diseases. And also the fact that we've already got a portfolio of candidate compounds that have shown very compelling data in preclinical models that are moving through into IND-enabling studies very well and position us to move into the clinic next year. So with the Series A, we're seeking to do two key things. We're seeking to bring our lead compound, TQS168, into the clinic and prove the concept clinically, which means two things. One is the obvious of getting early data in the target orphan neurodegenerative diseases we're positioning the lead compound for. But also, in addition to that, is proving the concept of this pathway that we're targeting. And that we can do that by having already an on-target clinical biomarker which supports the mechanism of action and can track disease. So those are sort of the two things we're seeking to do in terms of development and clinically. And then upstream, we're seeking to bring forward the next generation of compounds and position those for the broader, more prevalent neurodegenerative diseases such as Parkinson's and Alzheimer's, as well as expanding our capability and our focus across these very interesting pathways targeting myeloid immune cell dysfunction.
Right, and the approach is to actually sort of activate these pathways rather than sort of replace sort of your molecules that are normally missing.
Exactly. We're seeking to correct the defects. So we do that at a pretty fundamental level where we upregulate the target pathway to restore, to reduce the inflammatory response of the cell, in other words, the pathological response, and restore the normal physiology of the cell, in other words, to restore its normal function, performing a cleanup of dead, decaying, and toxic matter around neurons.
Yeah. So looking at the sort of the patent claims that are associated, with the molecule and what came out of the Engelman lab, it looks like those claims sort of are saying that it could be used to treat a number of different neurodegenerative diseases. Which ones are you going to pursue first, and why?
Yeah, that's a good point. So this is the sort of the luxury of choice, I suppose. We have too much choice in some respects, but we're very focused. So this is a common pathway, and it's one that is a powerful and early pathway we've identified in multiple neurodegenerative diseases. Hence the fact that we have coverage over a wide range of therapeutic opportunities. What we're doing as a company of our stage of development, we're focused initially on going further and further downstream in development into the clinic and then into later stage trials in orphan neurodegenerative diseases. So the lead compound TQS168 is positioned for diseases such as ALS and frontotemporal dementia, it may well be applicable in other neurodegenerative diseases as well, particularly the orphans that we're strategically reserving for that compound. And then what we'll be doing is bringing forward the next generation of compounds for the broader neurodegenerative diseases as well. And we have a broad patent estate. We've already got issued patents, the ones that you're already referring to, but we also have patents that are going through prosecution right now, very, very well, that give us very broad coverage over what we're doing, both in terms of use and composition of matter.
All right, so these patents are issued. So has Tranquis got exclusive licenses to them? Because I'm assuming that Stanford was there in the first place.
Yes, we exclusively licensed all the technology underlying these programs from Stanford in 2017. That's when the company got going. And then I came on board in 2018 to take it forward. But we have exclusive rights over all of this. And there are additional patents that were issued by Stanford that we continue to progress and give us even broader coverage.
Right. We haven't actually mentioned who your investors are. So do you want to give a shout out to the people who were, I guess, were involved in the seeding in the first place and who's now participating in the Series A?
Yeah, so the seed was led by SR1 and the founders and a couple of their friends. It was a very supportive and small group of investors there. It's unusual for SR1 to be doing seed rounds, but they really wanted to get into this very early on. And SR1 brought me in, Raji Dadu, who sits on the board of Tranquis, a very senior partner at SR1, was also on the board of my first company and invested there through SR1 as well. So that was the seed round that enabled us to get the company going, spin out from Stanford, continue to do the animal model work, and start to broaden out our portfolio. And then the Series A that by the time this goes out, we would have announced, it's just coming up, $30 million, and that's being co-led by Revolutionising Ventures, which is a new up-and-coming Japanese American fund with a phenomenal focus in metabolic and immunometabolic diseases as well as neurodegenerative diseases. So we really liked their understanding of what we were doing. They co-led that with SR1, and then there's participation also from Vivo Capital, Correlation Ventures, and a new fund called Hillsborough Venture, amongst others.
Right. Okay. So, you mentioned that you're hoping to get the lead compound into the clinic, etc. What are the sort of the next key milestones that we can expect to hear from Tranquis?
The next big one that you'll hear is we intend to start clinical studies with our lead compound next year. So that would be the next one that you should look out for, and obviously Mike will keep you informed. And then beyond that, we'll obviously be seeking to announce early data in healthy volunteers, and then potentially also in patients as well, and in particular, work on the biomarker. That's sort of the progression downstream in development. And then we'll be announcing developments in terms of our overall research strategy, next-gen compounds, and our approach towards these targets in due course as well, and in parallel with some of those big clinical milestones that we've been discussing.
Right, and so the Tranquis business model, how far do you sort of anticipate taking these compounds, or are you going to be sort of getting partnerships from pharmaceutical companies if and when you can?
Well, we're very well supported to achieve a few key things in the next couple of years, and as I mentioned a little earlier, one of the big things we want to show is that this is a revolution in neurodegenerative disease management and treatment. By getting the product and getting the lead compound to a point where we can show its effect in patients with orphan diseases, but also show the underlying, prove the concept of the underlying mechanism. So extremely well supported to do that, as well as progress the upstream molecules and the research and science base. So that's what we're focused on right now. But as you can imagine, once we've shown that we're getting a signal in patients and that correlates with an on-target biomarker that supports the underlying mechanism, we see that there'll be multiple opportunities to take us forward through financing paths and potentially partnering paths as well, Some of those may come earlier or later, but we're really focused on that key inflection point. And once that comes, we'll be able to then pursue the next step with the company. At the moment, we're really just focused on getting 168 to the clinic and proving the concept over here.
Great. So Sanjay, look, it's been a pleasure talking to you again. So thanks very much for taking the time to speak to me today. And this sounds all very exciting. I mean, neurodegenerative diseases are, as I said, clearly a major medical need, and the approach that you're taking is going to be keenly watched by us all. So we wish you well with that effort. So if you'd like to tune into future conversations, follow our LinkedIn page where we'll be posting alerts to future episode releases. So in closing, I'd like to thank Sanjay again for joining us, and thank all our listeners for tuning in. So until next time, stay safe and healthy. I'm Mike Ward, and I'll see you in the next episode.
Thank you, Mike. It's been a pleasure participating, and thank you very much for those insightful questions.